Hello everyone, we are Ariana Panels and Xiaoyu Chen. Our project is about combining sample one clustering and orthologous gene identification for single cell RNA-seq data analysis. So why do we want to do this? Because computationally, as we learned in the class, there is not a sample one version of clustering algorithm like the gene sampling in multiple discovery that combines the advantage of hot clusters and being probabilistic, and also biologically, in an environmental or hospital context, the samples might be a mixture of cells from different species. So, based on that, we have three specific aims. So, firstly, is to identify orthologous genes shared between human and mice, and secondly, to design a sample one stochastic expression clustering algorithm for clustering single cell RNA-seq data. And third is to combine them to and apply that to a mixed sample of human and mouse. And the data set we use for this is a neural cortex single cell RNA-seq data for both human and mouse brain. And firstly, as for orthologous genes in human and mouse, we found that only a small proportion of genes are found with no orthologs. But those genes found with no orthologs are significantly more highly expressed than those genes not found with no orthologs, suggesting that these genes with common ancestors are more important in cell function.
So we designed this stochastic clustering expression algorithm in order to basically combine the benefits of having a probabilistic result as well as having hard clusters. So basically what we did was similar to the give sampling algorithm we discovered that we learned about in class. What we did is we took some of so we had expression data, and for each individual sample, we assigned it to an initial cluster, and then computed the averages for that. With each iteration of the algorithm, we took out a few of the points actually. So we started out just taking one, but because the data sets were so big, that caused convergence to take many, many iterations. So what we did basically was scale the number of points that we removed on each iteration of the algorithm proportional to how different the overall radius or the spread of the points in that cluster was from the maximum in the whole data set. So when we started out, we took if all data points were in the same cluster, what's basically the largest distance from the average of that cluster to the average expression profile to any point. And then based on the ratio of that, we scaled how many points we took out each time that we recompleted the averages and reassigned the points. And eventually, after a maximum number of iterations, we stopped clustering. So here's just an example of that stochastic expression clustering algorithm. So this is for just a simple expression data set using a heat shock experiment data set. And basically, here is just showing that it sort of reproduces the results of a normal k-means algorithm for a really simple PCA like this, where you don't have a lot of separation between clusters. With this is sort of a low criterion here, so you can see that some points are randomly assigned to one cluster or the other, so you have some on either side. On the right there, you see the radius of the clusters going down with each iteration, so that's just the distance, so that maximum distance from any point to the average center of that cluster. So you can see it decreasing over time as you would hope over the course of the iterations. So the benefits of this clustering algorithm are really just the modularity as well as the hard clusters being assigned as combined with the probabilistic approach. So basically the user can decide what the weights are that it wants to assign to the different features. So if you're using principal component analysis data, for example, you can create so I had a vector of weights basically that you were using to compute that average. And if you want the first principal component to be especially highly weighted for some reason, or if you're using other features of the data where you know some are more important than others, you can use that factored into your average expression profile and use that as a criterion for clustering. You can also modify how quickly you want it to converge or how much randomness. Basically, when we were deciding on clusters, there's a probability that it will be assigned to any of the clusters, and you can change the stringency of that requirement if you want tighter or less tight clusters based on your assumptions. So in order to sort of combine the stochastic expression clustering approach as well as our original work with orthology, we wanted to work with single cell data, and we wanted to specifically investigate whether we could make inferences on what would happen if we had a mixed sample single cell data set. So in this case, we used well-known data sets from the Allen Brain Institute single cell data sets for the visual cortex, And basically what we did was we combined the human and the mouse data and then noised the data as if they were combined, and we had some sort of mapping or expression errors between the two. So you can see those 0 matrices on there is what up there is what you start with. And then we simulated noise between them by basically adding randomness to those 0 matrices and measured the noise sensitivity. So the top two here, on the top left, that's noise being added to all genes. On the top right, that's noise being added to genes without orthologs. And the bottom two are both orthologs, add noise to genes that had no orthologs between the two data sets. And this is basically a metric of how tight the clusters are. So this Kolinsky-Vargas index score is basically a higher score, indicates more differentiated clusters. So the two clusters between the two species are tighter, so you have more differentiation between human and mouse. So we can see that the highest impact on the tightness of the cluster, so basically the lowest score, is at intermediate levels of sparsity, which corresponds to the actual sparsity of the original data set. So you have a sparsity of around 30% of the single cell expression data, and if you replicate that with the noise, then that's when you see the clusters being closest together. So it's hardest to tell the difference between mouse samples and human samples. But you also see very clearly from the top and the bottom, looking at the relative scaling of these, that if you're adding noise to genes with known orthologs, that causes a lot less differentiation of clusters, indicating that the high expression levels and the importance of these highly conserved genes with known orthologs in other species has more of an impact on whether we can separate out the two organisms. So basically the takeaway here is that using single cell mixed species data, there's considerations in terms of orthologous genes might be more important than non-orthologous genes. We developed the stochastic clustering algorithm to be able to handle situations like this. So when we're clustering those data with our stochastic clustering expression algorithm, we have more flexibility in order to be able to tell the difference between points with overlap. So in addition to being able to effectively reproduce conventional clustering, our stochastic expression clustering algorithm can take into account things like orthologous genes and single cell, mixed species single cell data sets that might be important in terms of separating out species identity, as experiments are being conducted now with mixed species data sets, such as for hospital data and the case of our research environmental data.
Great. If you go back to your count of orthologs versus non-orthologs, you were able to find, I mean, how many genes are there in the human genome?
That should be 30,000.
Okay, there are about 20,000.
Yeah.
Neural cortex.
Sorry.
So this is actually, this is because we use the annotation from single cell data sets. So these are not really genes, but it's because we use
Transcripts?
Yeah, so they're what was mapped to a part of the gene from that, like the single cell data set. So we use the GTF, so like basically it could be a gene region. And then we used an orthologous gene finding algorithm to basically, so we pulled out using
because there are maps of orthologs between human and mouse.
Yeah.
You could just start with those genes and map transcriptomics to those genes. There's about 20,000.
Yeah, so we wanted to have it be, so like looking ahead to doing the single cell part, we wanted it to correspond to the columns of that matrix. So that's why we used this approach and just like took the human genome and used that reference to the GTF file that was associated with that, and then pulled out whatever it said for genes and then mapped those, so that we could then, when we were doing the single cell later part of the project, we used the same gene names.
Other questions?
It's
I'm a little confused. Can you go to the block matrix slide?
Yeah.
If you have genes that are orthologous between human and mouse, why would you like stack the orthologs? Why are they?
Yeah, so those are they're orthologs, but they're genes with known orthologs in the other. So like this, you wouldn't have any counts in the mouse data for the human name of that gene, even though they have orthologs. It's like it has a different name in the two, or it has a different column in that original data set. So you could theoretically combine those counts for doing one of those experiments, but because we didn't have data from an actual mixed sample experiment, those were separated out.
And then how are you adding those?
So it was based on, so like we had either orthologs or non-orthologs. We used basically the average expression over all of them, or we used, so in the case of when we were doing this metric, you can see that that is like a proportion of non-zeros, it's like a sparsity, and then generating random numbers from that, and then like, so generating random numbers within whatever range was appropriate for that class, based on the average, centered around that average, and then modifying it by whatever that sparsity was that we were looking for. Yeah.
So for the folks who need to leave early, maybe now's a good time to do that. So who needs to leave the earliest? Okay, why don't you guys come? I'm sorry. Oh, you will leave? Okay.
Yeah.
So you're next?
Yeah.
Okay, go ahead.
Over. I got these one. We're a little far into our presentation.
Sorry, are you, this is your slide?
This was like slide number 3 of our slides. Hold on.
Oh,
Here we go.
yes.
Okay, we're using.